The cell cycle is a regulated sequence in which a cell grows, duplicates its DNA, and prepares for cell division.
The main cycle consists of G1, S, G2, and M phases. Some cells can exit the cycle into G-row, a quiescent state. G0 is a quiescent state outside the active cycle. Cells remain functional, and some can re-enter G1 when appropriate signals stimulate proliferation. During G1, the cell grows, synthesizes proteins and organelles, and prepares the machinery required for DNA replication. During S phase, the cell replicates its nuclear DNA. Each daughter DNA molecule contains one parental strand and one newly synthesized strand. During G2, the cell continues growing, produces proteins needed for division, and checks its replicated DNA before entering M phase.
Interphase includes G1, S, and G2. It is an active period of growth, DNA replication, and preparation before cell division. Cell cycle progression must be tightly regulated. Control mechanisms prevent damaged or unprepared cells from progressing to the next phase. Cyclins activate cyclin-dependent kinases, or CDKs. These complexes phosphorylate target proteins and drive the cell through specific stages of the cycle.
At the G1S checkpoint, the cell assesses growth conditions, available resources, and DNA integrity before committing to DNA replication. Early in G1, RB restrains E2F. Cyclin D CDK4/6 phosphorylates, releasing E2F to activate genes required for S-phase entry. DNA damage activates P53, which induces P21. P21 inhibits cyclin CDK activity, causing cell cycle arrest and allowing time for repair. At the G2M checkpoint, the cell verifies that DNA replication is complete and that the DNA is sufficiently intact before entering M phase. At the G2M checkpoint, the spindle assembly checkpoint ensures chromosomes are properly attached to spindle microtubules before chromosome separation begins. At the cell cycle control is disrupted, damaged cells may continue proliferating. Persistent dysregulation can contribute to uncontrolled cell growth and cancer. Many anti-cancer therapies exploit abnormal cell proliferation. Some target DNA synthesis or mitosis, but rapidly dividing normal tissues can also be affected. Ki67 is a nuclear marker associated with actively proliferating cells and is widely used to assess proliferative activity in tumors and tissues. In oral squamous cell carcinoma, alterations in cell cycle control can disrupt normal proliferation and differentiation, contributing to uncontrolled tumor growth. The cell cycle coordinates growth, DNA replication, and division. Through cyclins, CDKs, and checkpoint, cancer can result. Next, we'll explore mitosis and meiosis in dedicated videos. Subscribe to Dentistry Simplified 31 for more high-yield dental and medical education.